Welcome, everybody. Zoomers, you are muted. Please stay muted. At the end, with questions and answers, you can type in the chat your questions and we'll get the speaker to answer them. If you have a cell phone in the room, if you would please silence it, I'd greatly appreciate it. Hopefully it won't go off. And Mary Chrisman is going to introduce our speaker for today.
Okay. Hello, everyone, and welcome to the third lecture in our Evolution is Everywhere course. And so today we have Dr. Joel Brown. He's the senior member of the Integrated Mathematical Oncology at the Moffitt Cancer Center and a distinguished professor of biology emeritus at the University of Illinois, Chicago. He's a wildlife biologist, an urban ecologist, and a cancer researcher. I mean, a little peripatetic there, you know? Childhood experience in Zimbabwe created a love of nature and animals that grew into an academic career. For 30 years, his lab and graduate students have worked from A to Z, aardvarks to zebras, with a particular love of squirrels. I'm not sure about that one. He takes an evolutionary approach to wildlife behavior, population, communities, conservation, and most recently, cancer, which is today's talk. He applies ecological and evolutionary principles to define, study, mathematically model, and treat cancer. With colleagues, he's developed the first mathematical model of adaptive therapy that was applied to a successful clinical trial of castrate-resistant metastatic prostate cancer. He's completed his bachelor's degree in zoology at Pomona and his PhD in ecology and evolutionary biology at the University of Arizona. So please welcome Dr. Joel Brown, who will today be talking about cancer and evolution. Thank you.
Okay, can everybody hear me okay? Is this just the right level? Excellent. Mary, thank you so much for the invitation. The opportunity to be here is really wonderful. I did notice you came up as Merry Christmas in the translator up there. So I think that says a good thing about you. You truly are a gift to all of us, so thanks. Also, I want to thank Lynn Holt, her husband, Bob Holt in Ecology and Evolution at UF. Was actually the outside committee member of my PhD committee back at the University of Arizona. So if you want really kind of a small world. And And as you can tell from all the things that I've worked on, don't let the secret out. I'm really a dilettante, not necessarily a serious scientist on anything, but I do have to ask, because I do love squirrels, how many people in here like squirrels? Good. How many people have a love-hate relationship with squirrels? You bet. The great thing about squirrels is every one of us has a squirrel story. But today's story is actually going to be on cancer. And my background is as an evolutionary ecologist. That means much of my career has been spent studying behaviors, ecological communities around the world through the adaptations of the organisms. And in this case, I bring the same kind of approach then to cancer. How did I get into cancer? Well, my game theory mentor at the University of Arizona, he was my co-advisor on my PhD committee. He began to work with a radiologist who's head of radiology at the Moffitt Cancer Center, Bob Gatenby. They kept inviting me to work on cancer. And then in a moment of weakness in Poland in 2008, agreed to work with them. Tragically, Tom Vincent died of pancreatic cancer the following October. And that brings us to the notion then of, what do I do now? So I'm actually have the pleasure of being part of the only department anywhere in a cancer center, a university, anywhere in the world, in which we bring together mathematicians specifically with the goal of bridging cancer biology, modeling, and therapy. So under the auspices of Sandy Anderson, our fearless leader in the department, there's 9 faculty members, and there's about 30, 35 postdocs, grad students, and other staff folks. What do? We do just that. We use mathematical approaches, but specifically to see if we can bridge mouse models, cancer biology, but ultimately, can we lead to better therapy and outcomes for cancer patients? Why? Well, if you go way back, and this is the nice thing about talking to this group, because I remember as well, how many of you remember putting a man on the moon? Yeah. How many of you remember, of course, the whole technological progress of basically high-speed trains? And of course, all the things. How many would be lost without their iPhone? How many people remember a rotary phone back in those days? And the check was in the mail. So obviously, there's been huge progress since then. But when it comes to cancer, here's the imperative. So that's the death rate of folks in the United States by various disease categories. And what you'll notice is the good news is that in general, life expectancy in the United States is going up. COVID, of course, was an exception. But you'll notice that the big killer is heart disease. But notice since 1930, look at the amazing progress that's happened in heart disease. In fact, look at about all the other ways to die. They have gone down. But when it comes to the likelihood of dying of cancer, the likelihood of cancer being part of the cause of death on your death certificate, what do you notice? It's kind of a flat line. And that's despite over $1 trillion, some of the very best minds in the last 10 years. And so in the case of cancer, while there have been huge improvements, we'll talk about some of those, overall, we've not kept up with heart disease, have not kept up with all the other ones. So the imperative here is the fact that every one of you in this room, I suspect, are either cancer survivors, have had suffered cancer, or certainly had loved ones, family members, friends that have died of this horrible disease. So why have we not been successful with cancer? So over this 50 years, 60 years since the war on cancer, of course, we had the cancer moonshot under Biden. The simple answer is evolution. Most cancers, almost all the time, would be easily cured, except that cancer cells evolve resistance to the drug therapies, whether it's radiotherapy, whether it's chemotherapy, immunotherapy, you can name it. So evolution by natural selection is what poses the biggest challenge to being able to successfully cure and manage, particularly late-stage cancers. So what I want to do today, I'm actually going to talk about what is cancer, and I would like you all to think about for a moment, how would you define cancer to a 16-year-old? Then I'd like to talk about cancer as kind of an ecological and evolutionary process. And then I'd like to talk about kind of new developments in therapy that we refer to as evolutionary therapies. So here's the standard definition of cancer, and this might be the one that you had in your heads. And that is, cancer is a disease of uncontrolled proliferation, basically by cells that are behaving badly that have the capacity to invade other parts of your body. This is the definition that's at NIH, NCI, the Swedish Cancer Center, the Chinese, you name it, Oxford English Dictionary. But I'm going to suggest to you that this definition may be part of our problem. It tells you what cancer does, but would you agree it doesn't really tell you what cancer is? Still doesn't tell you what cancer is. And so my role in the field has been to, part of it is see if we can think about cancer in a different way and if that can help us. So I would like you to imagine cancer as a speciation event. So imagine that it's a cell of your body that jumps levels of natural selection. What does that mean? That means originally the cell in your body was part of you. It was part of you as a whole organism. It was part of you as a whole organism trait. But now, through a variety of unfortunate genetic processes, it now becomes your own personalized pathogen. It now becomes a new single-cell beast. The tumor becomes its ecosystem. It's unique in the sense that you are its planet Earth, as opposed to infectious diseases that are bouncing from person to person. And of course, it ends with the patient. So with that in mind, got together with a group of people. First off, whenever you think you have a cool idea, the cancer is a speciation event, and you start digging around in the literature, you realize I'm not unique. It's not original to me or to several other people. In fact, it actually goes back into the 1950s to Julian Huxley, who was actually an evolutionary biologist who brought on, for those that are familiar with the modern synthesis, the synthesis of genetics with evolution by natural selection. You'll notice back in 1956, he proposed this. So we have recently, with this team of folks, fine scientists from a variety of places, Princeton, Moffitt, Johns Hopkins, this is kind of our new updated definition of cancer. We tried to retrain as much as possible that which is familiar to cancer biologists and to oncologists. That's what they learn in med school. So now I'd like to leave you with the idea that cancer is a disease of uncontrolled proliferation by transformed cells, meaning, yes, they've undergone a transformation. But the key addition, and this, of course, is what's going to motivate our talk today, is that they are subject to evolution by natural selection within you. They are subject to evolution by natural selection. Hence, then, cancer becomes pretty much a disease of fighting evolution by natural selection. So, cancer will be the most personal experience any of us will ever have of evolution by natural selection. It will also explain why it's challenging to treat and what we run up against. So, I'd like to point out a few things about cancer. Like life in general, because it has then similar properties, is it's a complex dynamical system. Secondly, it's hierarchical, all the way from molecules up to ecosystems. And as I said, in the case of cancer, it's we, the patient, as the planet Earth, and the tumor as its ecosystem. But I want to focus on the fact that it is the product then of evolution by natural selection operating within us. So a real quick run through. I know you've had a number of talks on evolution, so you probably have seen this, but I'll go through it quickly to point out that cancer satisfies all of the properties we need for evolution by natural selection. So first, there's heritable variation. Heritable variation just says like begets like, but with some boo-boos. So for instance, your children, your relatives, your parents, they kind of look like you, right? They kind of look like you. And of course, if the kid doesn't look anything like father, well, there's always the postman, of course. Or in my case, I have two lovely daughters. Thank goodness my genes are recessive. They look just like their beautiful mother. But like begins like with mistakes, and indeed with cancer cells, that is very true. In fact, heritable variation in cancer is terrifying. Not only do cancer cells generate heritable variation by mutations, the classic genetic mutations, guess what? They have access to your entire human genome. They have the entire human genome. They can go around with epigenetic bolt cutters, and they can open up all sorts of cassettes of genes. Furthermore, they get copy number variation. They also get chromosomal rearrangements. Their karyotypes don't look anything like what your karyotype originally looked like. And here's what's really scary. They even have parasexual reproduction. Cancer cells will fuse and combine their genetic material. Furthermore, they will undergo polyploidy, aneuploidy. So we now know, boy, the ways that cancer cells can generate heritable variation is truly remarkable, amazing, and scary. The second attribute of evolution by natural selection is there must be a struggle for existence, and that is organisms have the capacity to produce way more offspring than can possibly survive. That's also true of cancer cells. It's totally terrifying to imagine that a rapidly growing tumor, a glioblastoma in the brain, can be expanding at 4% per day. Even a slow-growing prostate cancer can grow at 1 to 2.5% per day. Pediatric sarcomas can be as high as 8 to 9% per day. But guess what? The actual capacity of a cancer cell to grow is 70% per day. So they have a struggle for existence. Within the tumor environment, they run out of space, use up nutrients. They do have to deal with the same kind of ecological problems that any organism does. And then the final bit is the heritable variation influences the struggle. That within these chromosomal rearrangements, these epigenetic changes, they have better ideas. They have ways of being able to compete better with each other. They have ways to invade other parts of your body. And they also have ways to be able to expand the boundaries of the tumor. So in a sense, cancer cells exhibit like what you can see up here. How many people in here are dog lovers? Oh, very cool. How many people like little dogs? Okay, how many people like the big dogs? At any rate, one thing is clear. When you look at everything from a teacup Chihuahua to a Great Dane, does it look like a wolf? No. Does a wolf even know that that's part of its inner wolf? I suspect most wolves would be embarrassed to know that a teacup Chihuahua. Could you imagine a wolf looking at a pug and saying, how did we spawn that? So of course, what happens in nature is you have heritable variation, and then that heritable variation matches the environment. And then what emerges then are the successful phenotypes. We see that with domestic dogs. And so at the end of the day, what we see in nature and what we see in cancer is actually a rather dull subset of what is evolutionarily feasible and what is ecologically acceptable. And my job as an evolutionary biologist then is to study what is, but also to study evolutionarily what could be. And the reason for that is how can we then understand and trick cancer? So my favorite area of mathematics is game theory. How many of you saw A Beautiful Mind? Sure, very cool. John Nash, wonderful aspects of game theory. So basically, we all are game theorists. And that is game theory deals with any situation where your best behavior depends upon the behaviors of others. So go back to your childhood. How many of you had strategies for manipulating your parents into saying yes? How many of you had parents that were on to you? So that's game theory. In fact, we're all practitioners of game theory. What do games have? They have players, they have strategies, they have rules, and they have payoffs. What does it mean for cancer to be an evolutionary game? In this sense, the cancer cells are the players. Their heritable traits are their strategies. Their payoffs are their proliferation rates, their survival and proliferation, and then the tumor microenvironment, the circumstance within which they live in the tumor is what sets the rules. So there's a number of key questions that I would like to invite you to think about in cancer. So we know that cancer is very heterogeneous. We know that there's lots and lots of genetic variability within cancer. Anybody that comes in and is tested, they will test for that. But we know in nature, when you go out there, how many bird watchers do we have here? Do we have any bird watchers here? Very cool. And of course, the fun part is seeing the spring migrants, as I'm sure have been coming through or going through, and to see the diversity of birds, all with their different lifestyles, right? So an interesting question then is cancer cell just a big pool of variation, or do they fill niches? Do they show the same properties like birds do on this property? So one of my grad students, we wanted to look for whether we could identify different species of cancer cells inhabiting tumors. We started with breast cancer, and I'll give a couple of examples from breast cancer. And in the breast cancer, we were interested in the idea that on the edge of the tumor, cancer cells are much more exposed to the immune system, to threats from immune cells, but it's resource rich. If you're in the interior of the tumor, it's resource poor, but you're farther away from the immune system. And we know in nature, food safety trade-offs are universal. Look at a porcupine. It doesn't have quills because it's a better way to get food. It has quills to avoid becoming food. And so you'll see organisms that emphasize safety, and you'll see organisms that emphasize competitive ability. So we want to ask the same question. In cancers, there's two really nerdy carbohydrases that are upregulated in cancers, been known since the 1950s, and that's CA9, CA12. So all you need to know about CA9 is CA9 creates a protective moat around a cancer cell that keeps away the immune cells. CA12 is a buffering agent that in the environment increases the flow of nutrients to the cancer cell. It makes you more competitive. So what we were interested in is, do cancer cells, do they all upregulate it? Are they able to combine being both a porcupine and being a deer, being a porcupine and being a rabbit? Or like rabbits and porcupines, you get distinct species. And what we found in every one of these women, 15 women, we sampled the edge of the tumor, we sampled the interior of the tumor, and in all cases, we found that 90% of the cells on the edge exhibit an anti-predator behavior. 90% of the cells in the interior exhibit a competitive behavior. We find the same species in tumors as we would find in nature. And it showed up in a whole bunch of different markers that we can use. And in fact, as a consequence of this work, there's now much more increased interest in CA9 and CA12 as histological biomarkers. And so up here is showing what we found. The yellow cells, this is just one example from one woman. The yellow cells then are the porcupines, and then the cells in the interior are more highly competitive. And notice the green ones are the only cells we found that upregulated. They just don't. So we find niche partitioning. Now, my academic granddad, Robert MacArthur, in fact, Bob Holt knew Robert MacArthur well. I never knew him. He died of cancer. But he was famous for showing that at small scales, you can get variation in different cell types. He showed that with warblers and showed how different species of warblers actually divide up the tree into different niches. So we were interested in whether we could find the same thing, again, looking at breast cancer. So anybody that's been to the desert or thinks about desert is you get what are called riparian habitats. So you can see this wash going through the desert. There's more water, there's more nutrients, and notice you get much thicker vegetation growing along here and sparser vegetation out there. Furthermore, not only is there thicker vegetation here, they're different species. They're not the same species. They're mesquite, they're ironwood. And here it's sage and creosote. So we wanted to see if we could find the same thing in cancer. Could we find this microhabitat variation? These are blood vessels. These are blood vessels. It's a 3D world. And what you'll notice is around the blood vessels, the cells are much more tightly packed than away. Furthermore, from the staining, you can tell there's two completely different types of cancer cells. Can you guys see that? They're not even the same. So as you move away from this blood vessel, it's like moving away from the dry wash. You not only see a thinning out of the cancer cells, you get a completely different type of cancer cells. So what that means then is when you're treating cancer, you're not treating a cancer cell. You're not treating, quote, the cancer cells. You're treating an entire community of household pests all at once. And does this then explain some of the challenges in therapy? So for instance, if any of the women in here who have had breast cancer, colleagues, friends, family that have had breast cancer, one of the first things they do is identify whether you are ER negative or ER positive. Basically, ER negative describes cancer cells that have freed themselves from the need for estrogen. Estrogen positive means the cancer cells require estrogen to be able to grow and to be able to proliferate. Now, with estrogen positive cells, you have the choice of using hormonal therapies. They'll use aromatase inhibitors, tamoxifen. There's a variety of drugs that can be applied. Now, what they don't tell you necessarily, although it's worth asking, is that every breast cancer patient will have ER positive and ER negative cells. It doesn't mean all the cells are ER positive. It doesn't mean they're all ER negative. In fact, in the patient I'm showing you here, the pink is normal cells. The green here are ER positive cells. The purple here are ER negative cells. So here's a patient that would be scored as ER positive, 22% ER negative cells. This patient would be considered ER negative there. And if you present different hospitals in the United States, or even different countries, will have a different cutoff for when they decide the patient is ER negative or ER positive. And that determines the therapy then that that particular woman will get for her breast cancer. So we were interested in the idea of, well, why do you get something like this versus something like that? So estrogen comes through the blood. Same sort of idea. We saw that riparian habitat. Estrogen comes through the blood. So maybe ER negative cancers tend to have less vasculature. Maybe there's less vasculature, less estrogen, so you favor a higher population of ER negative cells versus that. And indeed, that's what we found. So in this graph over here, this is mean vessel area, mean vessel perimeter, and taking then 20 plus women, taking women over 20 women and having them scored, how they were scored for their disease, we found that the women that were scored as ER positive had over twice as much vasculature in their tumors than the women that were scored ER negative. It makes sense ecologically. Now, furthermore, when you're giving therapy, how does the therapy get to the cancer cells? How? Through the blood. Through the blood. And we know that if you have a diagnosis of ER negative, that's considered to be a poor prognosis. And indeed, there's good reasons for why it's a poor prognosis. Much is understood. But if your drugs are being delivered through the blood, if ER negative cancers have half as much vasculature, what does that tell you about drug delivery? So for the same amount of drug, it's just not getting there. So interestingly and importantly, the challenges of treating ER negative breast cancers may also have to do with drug delivery. It may be just simply a property of the ecology of them. So as a new species, if we consider cancer to be a new species, what can we learn about it? Well, first, it's unusual. A multicellular organism gives rise to a single cell. The big speciation event is what we call polyphyletic. It's unique across every cancer patient. All of the millions of new cancer cases that will occur worldwide, each one is a unique event. We start to see what we refer to as an adaptive radiation. You see an adaptive radiation of the cancer cells as they fill different niches. And then like island adaptive radiations, think of all the islands in the Pacific that have come, created vast floras and faunas, and then disappear beneath the waves and are gone forever. In a sense, cancer has that property as well. Also, what's interesting is we generally in cancer biology compare cancer cells to normal cells. That's probably the wrong comparison, because what's abnormal for a cancer cell compared to a normal cell is not abnormal for a single-celled organism. And what looks like a totally complex tumor environment compared to the normal tissue actually looks pretty much like a standard ecosystem. So through the eyes of ecology and evolution, cancer suddenly doesn't seem quite as mysterious. Most of its properties, its evolution, its environment makes sense. So now you can start to see the next step of therapy once we view it as an ecological and evolutionary process. And that is therapy, trying to kill the cancer cells, basically becomes what we call a predator-prey game. It's a pest management game. And what's interesting as a pest management game is that it falls into the rubric of what we call leader-follower games. I'm sure you have heard all over the place of personalized medicine, the patient-specific care. They'll advertise, you hear it on the radio waves, we'll treat your patient just like you do as a unique family member. What that means is they measure the circumstances of the cancer very, very carefully. They apply the perfect therapy for where the cancer is, but then of course the cancer evolves. So wittingly or unwittingly, the moment therapy is applied to the patient, it's game on. If you do not kill them all through surgery, radiation, adjuvant therapy, if you do not kill them all and come out as a cancer survivor, the cancer cells will evolve, and what evolves is worse than what you started with. Does anybody remember the movie Gremlins? Yes, so anybody that remembers Gremlins, yes, that drop of water, that drop of therapy can open a Pandora's box. So I like to think of squirrel hunting as kind of the Stackelberg game, this leader-follower game. And going back to dogs, I'm sure you guys can relate. So here's a dog chasing a squirrel, chases it up a tree. Now, most dogs, not all, some people have very clever dogs, but most dogs, they point right at the squirrel. They chase right at the squirrel, and of course, what does the squirrel do? It runs up a tree. And then you see the dog go in a wide arc. It goes in a wide arc to chase the squirrel up the tree. Now, if the dog is fast enough, if the squirrel is far enough from a tree, then the squirrel is dead. Therapy works. Therapy works. However, if you're a coyote, what do you suppose a clever coyote does? Anybody want to suggest? Or a clever dog for that matter? It anticipates the tree that the squirrel is running to. It cuts it off. It cuts it off. Interestingly, most cancer therapy now, I hate to put it this way, is like the dumb dog. We measure exactly where the cancer is. We measure every property of that cancer down to the nanometer of where it is. We then blow up that spot for months, maybe a few years. And if at the bottom of that hole we cause in the patient, there's a dead squirrel, we won. But guess what? Much of the time, the squirrel hasn't been there for a long time. It's evolved and run up a tree. So the first step of evolutionary therapies is to try to anticipate where the cancer is going to go, not just where the cancer is now, but in response to therapy, where might the cancer go? We call that a resistance management plan. Have a plan for when it evolves. Furthermore, what is just beginning to happen, I have a patient that I've worked with, a number of patients who are now operating under this opportunity. Guess what? The squirrel may have multiple trees it can run to. Which tree is it going to go to? Similarly, with cancer, there are multiple resistance mechanisms. When you apply a therapy, for lung cancer, for instance, there are a variety of ways by which the cancer cells can thwart the therapy. Can you know that in advance? Does the squirrel even know in advance which tree it's going to run up? Is it only the attack, kind of like Schrodinger's cat? We don't know, but what we do know, does anybody know what a clever coyote does if it's not sure where the squirrel is going to go? It'll do a fake. It'll do a fake, just like a good soccer player. It'll do a fake, get the squirrel to commit to a tree, and then go across. What we don't do much in cancer is when you present with cancer, often the cancer at that point can be and may well turn out to be lethal. But for the next few months, perhaps even for the next few years, it is not lethal. You have time. You have time to explore. And so we have several patients now that are willing to audition the drugs, just like the coyote. Give a very short 3-week course of the drug and see what happens. Just see what happens. And then file it away. Then audition the next drug. Start to learn about the cancer. Explore the cancer. Try to get a sense of where that cancer is going to go. But right now, under current practice, which is understandable, when you go in and you're diagnosed with cancer, it's like ants in my pants. You want them gone as quickly as possible and as many as possible. However, when you're dealing with an evolving disease, what gives you short-term satisfaction may be selling out the future of therapy. Does that kind of make sense? So what do? We have modeled, have a great team of folks, Katerina Stankova, and then Johan and Monica. They're in the Netherlands at University of Delft, Bob Gatenby, who's always been our thought leader. We've modeled cancer as a leader-follower game. And this then has led to some of the different approaches to cancer therapy. So right now, there's 2 general approaches. One is, you might call it the silver bullet approach, and that's coming up with new drugs, new ways to treat cancer with the hope that it will cure most cancers most of the time. Right now, the flavor of the decade is immunotherapy. You may have heard of CAR T-cell therapy, adoptive therapy. It's amazing. And indeed, these create amazing drugs. However, in clinical trials now, they're very expensive. They can run into the hundreds of millions, if not billions of dollars. And whereas in the 1970s, almost half of all clinical trials were successful, now it's approaching 10% or even less. So part of the reason why drugs are so expensive is you're paying for the one that succeeded, but you're also paying for all the ones that didn't succeed. The other approach to cancer therapy right now is combinatorics, combining drugs, trying to add drugs together, sequence drugs, and indeed, these have been very successful in prolonging life, increasing quality of life, but not necessarily curing any life. So what we'd like to propose is a third way. We refer to these as evolutionarily enlightened management strategies. Actually, Mary, if you were in the fisheries industry, you'd be very aware of this in the sense that fishing not only can crash the fish population, it causes them to, honey, I shrunk the cod. They actually evolve to be smaller. So this notion of trying to model and anticipate and steer the evolution of any kind of pest or resource has actually been going on in the fisheries management for 30 years. It's not new. It's been going on in pest management in agriculture since the 1980s. It's now going on extensively to prevent antibiotic resistance in bacteria. But interestingly, it's only just now entering the world of cancer. And that's because it requires appreciating cancer as an evolving disease. So we apply adaptive therapy. Bob Gatenby proposed this in 2009. And the idea then is, can we use measures of the patients? Can we use historical retrospective studies of similar patients? Can we use mathematical models in which we now try to, like the squirrel, can we anticipate and steer the evolutionary dynamics of the disease? So here are some of the conceptual issues when we try to design an adaptive therapy trial, and I'll talk about a couple of trials. First off, maximum tolerable dose, which has been sort of the standard of care, which is hit them as hard as you can without killing the patient, without too much toxicity, shock and awe. It's evolutionarily unwise because you're applying maximum selection force. You're maximizing selection for resistance. Second, when you're dealing with metastatic cancer, if you have stage 1 cancer, stage 2 cancer, surgery, other techniques, cure. They cure most of the time. They're amazing. But as soon as the disease is metastatic, in most cases, it is now incurable. And at this point, resistance will happen. The other things we need to understand, we've already talked about the community. What is the state of the community? How does the therapy disrupt their ecology? What is the cost of resistance? What can you monitor? And this bottom one is the one we're most excited about, is encouraging the field to have a resistance management plan in place before you even need it. So before the patient even needs it, have a plan in place. So these aren't these great? These are currently the 5 types of evolutionary therapies. I'm not going to have time to describe each one of them. Competitive release therapy has already been used and is being used. Extinction therapy is what we're currently working on. These, you can ask me about them later. They get me super, super excited, but that's where you're really trying to mess with the cancer cells. So I spent essentially my entire career trying to figure out ways to keep snow leopards from going extinct, to keep black rhinos from going extinct. And now my job is to find my diabolical evil twin, to use these same ideas to see if we can extinct the cancer cells. It's kind of fun. So competitive release therapy is very simple. The idea is that when you apply therapy, you have sensitive cells, they respond to therapy, that's great. But of course, you then select for resistant cells. And of course, the problem is the resistant cells, they're just that. You don't have a therapy for them. And so with the evolution of resistance, then you've lost control of the cancer. So the idea is actually quite elementary and simple. It can be used in ecology as well. If you've got the sensitive cells, the blue ones, and there's a resistant cell, the red one, you apply maximum tolerable dose, you get a nice big effect, but you're only left with resistant cells, and then you have no way to control the cancer. So the idea is, what if you don't eliminate all the sensitive cells? You know you can treat the sensitive cells on demand, but maybe you can use the sensitive cells as your therapy for the resistant cells. Remember the struggle for survival in evolution by natural selection. So as the cancer cells are competing with each other, there can be a cost of resistance. And so maybe the sensitive cells can be used as your control of the resistance. So the idea then is to apply therapy, stop therapy while you still have lots of sensitive cells, let the cancer grow back, and then hit it again. Let it go back. Can you make it a chronic disease? Can you make it something that you can contain on there? And so that was kind of the idea behind this clinical trial. Now, this clinical trial started back in 2014. This is the one that, along with Jessica Cunningham, we got to write the mathematical model. Jingsong Zhang was the very courageous oncologist to try this. Now, adaptive therapy is more widely accepted. It is not challenging if you have the funds and the patients to get an adaptive therapy trial through the FDA. But at the time that Jingsong was proposing this trial, it was radical. The idea that you would stop therapy when you knew the therapy could still be very effective. How's that for wild? You stop the therapy when you know it's still working, and then you're going to stop therapy with the goal of letting the tumor come back, of letting the cancer cells recover. How's that for wild? And getting this through was not as easy as it might be today, but thanks to Jingsong. The idea was this type of patient, and this is where you want no man to be. Some of you may have had loved ones or whatnot that have died of this disease. When caught early, prostate cancer can be quite curable, which is good news. But here it shows typically what can happen to a patient. Patient presents with high PSA levels. The hope is it's contained entirely in the prostate. That's great. And so that means here you see a radical prostatectomy. That should cure it. The patient goes home. The hope is they're cured. And notice 500 days after the initial diagnosis and about 100 days after the prostatectomy, PSA goes up. And then that's the oh crap moment. That's the moment when you realize it's metastatic. It's in the bone. At that point, once it's in the bone, if that gentleman does not figure out another way to die in the next few years, the disease is invariably fatal. Now, with androgen deprivation therapy, they can prolong and create a sustained, almost remission state. But you can see in this patient, a long period, and then it comes back. And then you notice the sequence of secondary, tertiary drugs in an attempt to control the disease. Each one buys very temporary relief. Can you see that U? But each one is temporary. You're now fighting evolution by natural selection. And then you get to the end of abiraterone as your last-ditch drug here, goes down, and then the patient then succumbs to the disease. So the idea was, and this has been known, is in late-stage prostate cancer, there's actually 3 species of cells. Remember our Darwin's finches? Remember there's 3. There's T+, just like estrogen positive. They require testosterone. There's T-, they're independent. And then there's these really sinister guys. We call them TP. Now, they're known by CYP mutation, and it's well known in men. And guess what? These cells evolve to produce their own testosterone. Worse, they leak the testosterone into the tumor, providing a public good for these guys. So the idea then is at this late stage, the opportunities for therapy are very, very limited. So the goal was, this is just to show you, for those that like mathematics, here was the mathematical model that underpinned this clinical trial. But then in this clinical trial, we had a very simple rule of thumb, is to administer abiraterone to knock down the patient's PSA 50%. So if you were a man, you presented, we knew your PSA level, we would knock it down 50% and then stop therapy. And then re-administer when the PSA recovered. So we had a group of courageous men that were willing to try this, knowing that, and when we first tried this, initially, oh my gosh, Jingsong, he is steady as a rock. Bob Gatenby and I, we had nerves. Because theory told us that we could knock it down, it would come back, and the drug will still work. But what if it doesn't still work? Nobody had ever tried it. This was all playing a hunch. This was all predicated that we understand evolution by natural selection. But maybe we misidentified the game. Maybe something else was going on. And so I don't think the men appreciated how nervous we were when they came back and we gave them the drug, it was like, please, please, please. How's that for a basis to treat your loved one? How's that for the basis to treat somebody who has an incurable lethal disease? Anyway, these men were very courageous. In the event, these were our control cohort here. This is showing their progression-free survival of the 15 men. This is the trial cohort. Anywhere you see red, they're on drug. Anywhere you see black, they were off drug. So the good news on this particular trial is that over this period of time, at least the men on the trial did substantially better. The original control group had an overall survival of 31 months. So from the time and all of these men are deceased. In the group of our adaptive therapy trial, these 16 men here, their median overall survival is almost 2 years longer, and 4 of the men are still alive. We call them our super cyclers. They just go up and down, up and down, up and down on the therapy. The other key element is these men were on half as much therapy. So it's the same drug, half as much, and it was calculated that saved $75,000 a year. $75,000 a year. So as a consequence of the success of this, there's now 2 Dutch physicians and a Dutch scientist that are conducting the same clinical trial as a phase 3 trial, which means it can become standard of care in the Netherlands, also in Australia. We also now have a much more complicated trial. So that looks really complicated. So we gained confidence. We gained a lot of confidence from the trial. And furthermore, we failed. These men still progressed. So we still failed, but we did an after-action report. We could start to understand why we failed. And now if you go back, is the only reason we were allowed to do this trial is because these men were over here. Why wouldn't you try something weird? By the time you're here, and so in that sense, then, that was why the trial was allowed to proceed. But how many of you would agree that might it be a good idea to do something here? Now, that would have been impossible to have done originally. But as a consequence of this trial and other trials, we were able then to put together this trial on the men that have castrate sensitive. They're in that long flat zone. And this trial is ongoing. Excited about it. We've accrued the men that we need in the trial. We monitor both their testosterone. We monitor both their PSA levels. We're modulating 2 drugs now. It's a fairly kind of complicated decision tree, the mathematical model. But the good news is, with this group of men, the number to beat is under current care. 46 months is the current expectation under standard of care with castrate-sensitive prostate cancer. These are our current group of 16 men as I speak. 5 have progressed. Right now, if they all progress tomorrow, we would be at 54 months. Right now, we're expecting we're going to be able to go at least 66 months or even go farther. So this one, we're just writing up this trial. We've reached the point where we know it's a success. We don't know median survival yet. We're waiting. Hopefully, it's a long time. We're hoping it's a long time. The men on this trial are on no drug at all. So bear in mind, standard of care now is to be on 2 drugs. One is a chemical castration drug, an androgen deprivation therapy drug that most men don't like. And the second one is an androgen receptor chemotherapy, hormonal drug. So in this case, these men have been on no therapy at all for half the time. Their quality of life is substantially higher. Half the time, and they've only been on both drugs on average 14% of the time in this particular trial. So anyway, so overall, then this trial, we think it's a success. We're getting ready to publish the next round of this trial as having beat kind of current standard of care. I believe I'm running out of time. How am I doing? I'm probably way... Okay, do I have 5 more minutes? Because this one we're really excited about is, okay, so you saw that these ones where we're trying to contain, that's a nice idea. In some cases, we've been relatively successful. In other cases, we have failed. We've not been as successful as we'd like to be. So extinction therapy is this particular dream. What if we currently have all the drugs needed to cure most cancers most of the time? Then the question is, how do we deploy them? It all comes down to deployment. This is our current kind of extinction therapy team. We have 3 physicians, Damon Reed, Dawn Jingsong, who you've seen before. We've got a couple of good mathematicians, Bob Gatenby, myself. We've worked out a number of the procedures that can be used for extinction therapy. I'll mention there is currently 2 trials going on with extinction therapy, one with late-stage triple-negative breast cancer, and the other with a pediatric osteosarcoma. So you might think of this therapy, the idea is kind of a kick them when they're down. So the goal is when you have the disease in essentially remission, but you know it's not cured. Now, many cases, earlier stage diseases, when it's in remission, it can be cured. It may be cured. But for these late stage diseases, they aren't cured. The physician knows they aren't cured. But all we say is we just don't know where the cancer is. So what we usually do is follow then, is it the Winchester or Oxford rules of boxing? Where after we kick it down to remission, we kind of back off. We either keep doing the same thing or we back off until it gets off the mat again. So the idea here is to fight dirty. If you have a drug you're going to use later, why not use it earlier? So why not sequence drugs? So to give you an idea of what this kind of therapy can look like, here's sort of standard of care. You can see that this is sort of a standard model that matches disease progression. You get a good response, and then all the cells are essentially then resistant. What I want to show you here is kind of a window. I don't know if you can click. Here, I'll let you. Can I click? I want you guys to watch this. So what's happening here is 2 drugs are being administered. 2 drugs are being administered. There can be resistance to each of them. But what we're doing is in this graph, we're simply shifting when the first drug, when you switch from the first drug to the second drug. And then what you'll see is between these 2 switches, if you switch too soon, you get resistance to both drugs. If you switch too late, you get resistance to both drugs. But watch, there's a window of cure. So we kind of call that our window of cure. Can you kind of see that? How's that for amazing? This is playing with evolution. And it looks like that window of cure is you should never use a drug any less than about 45 days, and you probably shouldn't use it for any more than 90 to 100 days. How's that for interesting? Before you want to kick them, before you want to kick them again. Can this work? Well, this already happens. This is already essentially practiced for a variety of diseases, and in which you actually get cure, particularly in the lymphoblastic leukemia. Cure is now affected by giving a whole sequence of drugs, changing them up frequently, and that elicits cure. And there's a variety of others. Christopher Gregg was our first extinction therapy patient. He's very kindly given us permission, so don't worry, we're not breaking any kind of rules of patient confidentiality. You can actually find some great YouTube videos from this gentleman. He's become a friend, a colleague. He's a neurobiologist at the University of Utah. He's currently 49 years old with a couple of teenage kids. And here was his challenge. He was diagnosed with stage 4 male breast cancer. Stage 4 male breast cancer. First, that's a very, very rare disease, and it's invariably fatal. Upon diagnosis, there's a single drug he would be given until it doesn't work, and then the prognosis is survival of about 18 months at best, about 18 months at best. And then this is what he was facing. So this is what the physician would be imagining for a patient like Chris, and that is treatment is palliative. You're not going to try to cure. Start with the least aggressive treatment to preserve quality of life. Treat with one drug until progression, and then try the next drug until you run out of drugs. I kid you not, this is what's out there for the therapy recommendation. Focus on drugs, this makes sense. Monitor disease activity using occasional scans. So the goal is not to be intrusive on the patient. You can understand that. So here's what Chris wanted. Here was his takeaway message from what he was interested in. At the time he started this, 2 young kids, he really wasn't interested in the palliative kind of approach. So notice the objective of the approach is to create a small, sparse, homogeneous population of tumor cells that cannot sustain. If that fails, the treatments are designed such that we switch therapies before resistance occurs, so switch frequently. We start with the most aggressive drug with the goal of reaching no evidence of disease. So his whole treatment strategy was quite different. And this was the original plan for him. He was diagnosed in September 2019. He and us mapped out a whole sequence of drugs that were switched up every 90 to 120 days. There's good news and bad news in Chris's story. The good news is he went clinically undetectable in January of 2020. He stayed clinically undetectable until March 2023. At that point, from my perspective and a number of us on his caregiving team, we failed. Apologized to Chris. Chris, on the other hand, no. He just saw we now switch in. We already had a plan. We had a plan for when this happens, and that was to audition drugs and currently go into an adaptive therapy. So I'm happy to report as we speak, he's still in great health. So now he's going on 6 years. 6 years with his disease. He's in great health. And right now he still has quite a few therapy options. So he's 49 years old, and we hope to keep this going much longer. As an actual clinical trial, Damon Reed and then my graduate student, Mariah Presley, we put together a model. I'm not going to go into the details of the model other than to realize that this model has 3 drugs. These M's are doses, 3 drugs, 2 resistance strategies. And this describes pediatric rhabdomyosarcoma. It's a horrible childhood cancer. Survival rates are less than 5% in this disease. For any parent, it's horrifying. So Damon Reed was able to use that model to develop 4 arms of a current clinical trial that's going on across multiple institutions in the United States. Here was a sample run of the model, conventional therapy, an augmented first strike, using that first, second strike, extinction therapy, and then adaptive therapy. When we ran the model for all the different possible patients we could imagine, this was the outcome. It appears that the first strike, second strike is your highest likelihood of cure. Standard of care is your least chance of cure. So as a consequence, this is now a clinical trial. It's ongoing. This clinical trial is ongoing. We'll wait to sort of see what happens. So what's happening now, there's a growing number of trials on evolutionary therapy. We're excited about it. There are now evolutionary tumor boards to evaluate patients that have no curative options to see if we can imagine through the eyes of evolution. Can we anticipate and steer? And right now, there are individual physicians and patients that are actually requesting this. There's actually demand coming from physicians themselves and from the patients. So what's the ultimate goal then? Is to use principles of ecology and evolution, patient data, mathematical models to anticipate and steer cancer's ecoevolutionary dynamics. And hopefully, this is just the beginning. Thanks very much.
Oh, believe me, they were in. So, questions? Oh my God. I'm getting there. Don't use that. Not to use that one. Here you go, Henry.
Wow, that is just incredible. I have a question. When you know, or when you believe that it's in remission, but you suspect it's hanging out someplace in some other organ and just ready to make its, do you have a good predictive model as to which organ it would be, maybe based on the lymphatic system or something? And when you do find it again, has it evolved to look like a different kind of cancer cell at that point?
So I myself do not have a model, nor is this something that I have direct experience with. However, the field has considerable experience. So in the field, there's extensive data that when the disease recurs, where does it recur? Much of the time, unsurprisingly, it recurs where it had been before, or it recurs in the tissue. So for instance, with women with breast cancer, where it's in the bone, invariably it recurs in the bone. However, there are other examples, and there's examples in a variety of these diseases where you can have a patient that colorectal cancer, complete remission, and then a MET shows up in the lung or the brain or the liver, that these METs will show up somewhere else. There's a lot of research going on by many, many fine labs on this question of these, quote, persister cells, dormant cells. What were they? How long were they there? Have they been there for years? Or were they just populations that have been knocked down, that have been remaining residual? The group out of Johns Hopkins and out of a number of other labs are showing that often when these cells do come out of this kind of dormant or this state of remission, yes, they are now a different beast, that they've actually been sort of incubating evolution. And that's actually really similar to invasive species ecology. You actually recently published a paper on kind of comparing it to invasive species ecology, where often with invasive species, the invasive species will get in, but it won't spread. There'll be a small population that resides in the area. And then we know in invasive species ecology will also often undergo then just enough evolution that allows it to break out and spread. Now, some invasive species spread instantly. Others can take a long time. So that's an area where many, many labs are trying to look into it. So I guess to kind of summarize, one, there's a lot of work going on to be able to detect the disease at lower and lower amounts. Circulating DNA, tumor biomarkers, and an active area that the field is doing. The second is then, how did they survive? Why did they survive? And when they do emerge again as discernible disease, how much have they evolved? Yeah, so thank you for that question. Yeah, but I think you're probably appreciating it's a very dynamic disease. Yeah, that.
Yeah, I have a sort of question comment thing. I was wondering if any research is going on in this area now. I had very aggressive triple negative breast cancer and had, adenomatous and cytoxin, the whole shamil. But I had done some reading on fasting as far as helping to knock down cancer cells initially. And so I fasted for a couple days, only had water or tea before my treatments. And it had spread to my lymph nodes. I didn't have any side effects from the drugs, which the oncologist was amazed at. And also, at least so far, I've been cancer-free. One of the residents there was appalled that I would even try this. Most of the studies I read were anecdotal and also done on dogs, at least with the master saying, no, they didn't get very sick. Oh, okay. I was wondering if any research actually is going on in this area for an initial knockdown.
Right, so first, very, very sorry. I'm sorry to hear of your disease, and I admire your courage and your approach to it. So the fasting and trying to use what are called metabolic strikes now, there is actually quite a bit of growing evidence to suggest that these ketogenic diets, metabolic strikes, particularly what you described, fasting prior to the chemo, there's good mouse model experiments to suggest that that can help, and it certainly doesn't hurt. Now, there's certain types of diets and arrangements of diets. Boy, if you have liver cancer, get fructose out of your diet. There's a variety of kind of, sort of well documented. And then there's a lot of this that right now you might call a little bit of folk, or a little bit of it's based on just sort of intuition or logic. But given that, particularly in your stage of cancer, often the cancer cells, their resistance strategy is to pump the drug out. They go hypermetabolic and they pump the drug out. And by your fasting, if you can get your blood glucose down just a bit, in the tumor, that can really drop the glucose and that can increase the potential efficacy. So if the discipline and the fasting is done in alignment with your therapy, I think right now there's no harm and it can do a lot of good. If you are interested, Chris Gregg, and feel free to email me or just look up Chris Gregg. He is the one I know who's put together the biggest compendium. He uses fasting, he uses metabolic strikes. He's put together a whole sorts of diet strategies and ideas. And I think in the field, this is gaining acceptance. But what hasn't been done is full clinical trials of some of these approaches. Wishing you the very, very best. Oh, yay. Oh, I'm happy to hear that. Yes, that.
You've given us a fascinating lecture, but I've got two, I think, very quick questions. Number one, is the reason that cancer evolves because it opens up recessive or dormant genetic sections? That's number one. And number two, are there any, I know you have all sorts of drugs, but are there any living things that actually predate on cancer cells?
Those are both great questions. So first off, yes, there can be genetic mutations, there can be, but again, what cancer cells generally do is they upregulate. It's like a rheostat. They downregulate, they repurpose, or they turn things off and on. Most of the subsequent evolution, there's a lot of debate and discussion. I tend to fall into the camp, but again, it's an open question, that most of their variation, once they're a cancer, comes from exploring the human genome and unlocking epigenetically all of those silent genes. That would be akin to your recessive, but in this case, all these silent genes, they've got the whole human genome. And
I said recessive, but I meant the silent genes, yeah.
yeah, exactly. So they explore these silent genes. I can go on and on over just the amazing genes that they find in the genome. Speaking to your second question, of course, the immune system represents an obvious predator of cancers. There is a whole field opening up, and there's some cool work going on of trying to embrace microbes and to enlist microbes that then do one of two things. They either create antigenicity and attract the immune system. Those would be viral vaccines and things, but actually some where they will go and specifically attack cancer cells. The risk, of course, is whatever can attack the cancer cell might attack you.
I wonder if you can design nano machines that would go in and attack the cancer cells specifically.
So that might be the next phase. Obviously, CAR T cell therapy is where you take your own T cells, you entrain them on the cancer cells, you create your own specialized bloodhounds, your own specialized ninjas, and then you re-inject them. That's sort of similar, but they're a living ninja. To be able to, there is a lot of use of nanoparticles to try to deliver drugs and whatnot, and they've had some successes, some mixed. That would be so cool if you could identify. Now, but of course, that would be a robotic immune system. That could be useful. That would be useful way outside. So please do it. I hear a Nobel Prize. Yeah, you bet.
I have a dear friend, a childhood lifetime friend who is up in Hartford, and he's just discovered that he has metastatic prostate cancer. His wife is beside herself, and is not understanding why there's all these delays in getting with the doctors and getting things started. I wonder to what extent your research is being communicated, broadcast across all cancer clinics in the country, or the world for that matter. And a second question is, I've read that Kennedy has cut off some cancer research funding. I hope that your efforts have not been affected by that.
Two quite diverse. So first, I'm very, very sorry for the friend. The accessibility of these adaptive therapies right now are really at the discretion of the physician. Some physicians aren't even aware. Other physicians are aware, but they prefer to stick to sort of standard of care, which is understandable. And then there's others, if it's raised, are willing to. And first off, as you probably have pointed out, boy, when you're dealing with breast cancer and prostate cancer, I know this sounds horribly sexist, but every physician will tell you is men are horribly non-compliant. Women are much more compliant. And we've seen that in our studies as well. But anyway, you can make of it what you will. But at any rate, so one is to get them into the physician. These papers are available. The person can email me. Right now, probably the physician that his entire practice now is adaptive therapy. He will do nothing other than adaptive therapy. Ken Pienta at Johns Hopkins University. So you can either email me. I'd be happy to put you in contact with either folks at Moffitt, but Ken Pienta, Johns Hopkins University, he's amazing. The other who's an absolute wizard of this is Dawn Leman. She practices in Oregon, and her entire practice is now built around evolutionary therapies. These are some really amazing physicians. It's not a bad idea to at least get a second opinion. You know, again, it's the patient, their physician, obviously these decisions on these types of diseases. But yes, these are available. The literature is out there. And to your second question, yes, chaos is reigning. That chaos is reigning. Moffitt, we have a hiring freeze right now in new folks. We had meetings at NIH where on Thursday, you're all packed and ready to go. And then on Friday, we get a notice saying the meeting is off and that the program director cannot tell you why. And a few courageous program directors have said, I'll set up a Zoom for you guys off the books as a friend, not as an official. So yeah, so there's an awful lot of chaos and fear right now percolating through what's going to happen in this kind of, and mostly right now at Moffitt, the actual direct effect so far has not been huge, but the indirect effect of fear and uncertainty is definitely probably right now the most concerning. I just sent in a grant proposal today. We'll see. I'm waiting for any rate. So don't get me going. Yeah, but thank you for raising that point.
We have one question from the chat, and it's asking, are both adaptive and extinction therapies evolutionary by your early definition of heritability and natural selection?
Yes, so we see the adaptive therapy is predicated upon the fact that evolution has occurred and that the evolution of resistance, at least in a subpopulation of the cancer cells, is basically rendering therapy ineffective. And so the strategy then is, can we make it more of a chronic disease that can be contained? Extinction therapy is predicated on the idea that if you are going to prevent resistance from even happening, you need to hit them hard and you need to switch quickly. So it's predicated on the idea that if you use the same therapy, even when it seems to be working for too long, if there are cancer cells present, they will evolve resistance. So extinction therapy is really hit them so hard and change up so fast that evolutionarily they're unable to respond. And that's to prevent, actually a concept developed by Bob Holt, Lynn's husband, and that is you're trying to prevent what is called evolutionary rescue. And so that term and that concept becomes key when you're doing extinction therapies. I hope that answered your question.
Their question. Let's thank our speaker once more for a very interesting talk. And next week, we're moving into a very different topic, and that's lack of evolution in the antique fishes of Florida, namely the gar and alligator gar, I think, and sturgeon. So a very different talk next week. Thank you very much for coming, and see you all later. Thank you.